I have the immense pleasure to announce this year's keynote speaker, a close friend and colleague, and somebody who I admire immensely, Professor Mark Daly. Mark is the director of the Finnish Institute for Molecular Medicine at the University of Helsinki in Finland. And he's also the chief of the ATDU unit at MGH and a member at the Broad Institute. Mark is a true pioneer in the field of statistical genetics, and he's amongst the most cited scientists in the field, and actually one of the top 100 most cited scientists of all time. And he was elected to the National Academy of Medicine in 2017. Mark will talk to us on the topic of genetic discovery and where do we go from here. And I'm excited to moderate the Q&A afterwards. So with that, I'm going to hand over the floor to Mark. And Mark, please take it away.
Great. Well, after hopefully a more than pregnant pause, thank you, Cecilia. It's a great pleasure to be here, and quite for me gratifying to see how much activity there's been in the ICDA since it was just a twinkle in our eyes 4 years ago. And I apologize to those of you who are hoping for a more exciting keynote speaker. This is somewhat what happens when you overreach and don't get the person with the most exciting, cutting edge science. You end up with me instead. So, but it is my pleasure to be here and discuss, what human genetics, has to offer still in 2022 and where we go from here. And so I think obviously, those of you of my age or older lived through a period of time where if you were working on Mendelian disease, you were having a remarkable revelation of technologies allowing us to find for the first time the molecular underpinnings of so many Mendelian diseases, while those same technologies and techniques and efforts were essentially delivering next to nothing, despite considerable effort on many of our parts during that same period. And how this was then reversed in the next decade with the advent of genome-wide associations following on the heels of the completion of the Human Genome Project. As we moved through the last decade, the questions turned much more to how we interpret and understand the biology of disease, which was really the point of the whole activity. And so in inflammatory bowel diseases, which is the example that I will use the most today, we had a wonderful time, running GWAS studies and deeper fine mapping studies over the course of a decade between 2007, 2017. And in one sense, this was an extremely successful activity, but in another sense, as those of us working on many, many other diseases encountered, the vast majority of genetic dissociations, while we were able to discover them quite readily, or we developed the techniques over this decade to discover them quite readily, provided only pointers to the genomic regions that often spanned many genes and had various unannotated SNPs in linkage disequilibrium with each other, but didn't really provide us clear evidence for the specific genes and mutations that would help us understand the underpinnings of disease. In the case of IBD, we were fortunate that there were quite a number of those GWAS associations from the early studies that did pinpoint directly protein coding variants, despite the fact that we didn't have the high resolution mapping at the outset and so forth. It was quite obvious what the responsible variants were. And this was because the GWAS actually pointed us to relatively low frequency and what in retrospect now appears to be quite strong effect variants associated to disease. And so as we turned the page to 2018, we first, as a community, started coming together under a banner called ICDA to ask originally a very, very simple question, which was that we had seen over the previous 2 decades tremendous progress in the discovery of genetic variants for complex diseases, where prior to that time, we had discovered next to nothing, or in fact, in many cases, absolutely nothing about the genetics of disease, despite recognizing the great heritability of most of those diseases. And the question really became then, were there fundamental methods or reference data sets or large-scale projects that would be in the spirit of the Human Genome Project, the HapMap, and some of these other foundational activities that led ultimately to the success in gene discovery that could be then employed or deployed to unlock this next step and to really unblock the slow translation of associated variation into molecular mechanisms, and ultimately medicines. And so the ICDA came together and began brainstorming in 2018, a small meeting in New York City, a larger meeting in Oxford, fun meeting we were able to host in Helsinki for those who were hardy enough to travel to the Nordics to enjoy our unexpectedly pleasant weather for a few days in May in 2019. And then the official launching of ICDA in September 2019. Now this is, we're several years past that, and hopefully most of a pandemic past. But it's been really an amazing ride to discuss and brainstorm and come back to the fundamental question, as I circle back at the end that we originally laid out, because I think we should keep in mind, and I'm sure we'll hear so much exciting science and insights into these questions, that the primary thing that we sort of gathered together and are still trying to answer and address is, how do we really get the biological insights and the actionable insights from the genetic activities of the last decade and a half that were the entire point or the entire promise of, even back to engaging in the sequencing of the Human Genome Project, why we are doing all of this. So, framed another way, and we've used these slides, many of us, for several years now, the challenge is quite simple, that there are tens of thousands of genetic associations. In fact, now well more than 100,000 if you mine through the GWAS catalog and the really nice tools at Open Targets and so forth, across many diseases, phenotypes, and now extensive molecular quantitative traits that are available to us. But the progress in discovery is vastly outweighing the progress in interpreting those discoveries. And so without the interpretation, the discoveries are simply a stamp collection and not really productive in the way that we need them to be. So how do we speed this up? Well, the first steps, have been, sort of nicely coming online in recent years, and that is with the increase in the scale of genetic studies, with the increase in the diversity of the genetic studies, and with the advent of better fine mapping algorithms and so forth, we are seeing not only greater numbers of discoveries, but also improved refinements of genetic variation implicated by GWAS, to the point where we can see enrichments such as shown on these slides here on the left. I don't think anyone is surprised, and certainly no one has ever really been surprised, that protein coding variation plays a massively outsized role in human genetics of disease, whether it's common or rare. And that's certainly borne out by the enrichment, ultimately, when you get down to high resolution mapping of genetic variation across many, many phenotypes. Furthermore, if you look at the preprint by a wonderful graduate student I've been privileged to work with, along with Hilary Finucane, Masa Kanai, he has done an integration of fine mapping results from across the 3 biobanks, Biobank Japan, FinGen, and UK Biobank, demonstrating that, in fact, not only can you occasionally pinpoint a protein coding variant in a gene, if you apply multiple diverse biobanks, you can often begin to flesh out immediately without any excessive effort or even deep sequencing, allelic series in genes that correspond to multiple independent variants, indicating the same phenotype as responsible. And so this meets and surpasses our old traditional Mendelian guidelines of 2 independent mutations in the same gene in 2 different families explaining the phenotype. And we can, harken back to those days and say, yeah, okay, we're now starting to, for at least a subset of our discoveries, to meet those stringent criteria. There's no need to rewrite them. They were good then, they're good now. But the challenge is, of course, that the vast majority of the genetic associations that are delivered by GWAS studies, clearly post-fine mapping, do not implicate a coding variant. And we can just say this is, roughly speaking, 90%, 10%. It depends a lot on the phenotype. It's not a constant number at all. For some, like our autoimmune and inflammatory diseases, the number is clearly higher than 10%. For many others, such as schizophrenia, and I'll touch on this later, the number is clearly definitively lower than 10%. And for simpler, rarer, and high penetrance diseases, the number is obviously likely almost reversed in this case. So one of the challenges, and I think sort of the way that many of us have originally formulated the thinking around ICDA, and to an extent, I think this is still a very important and obviously massively, still yet to be understood vista in genetics and biology, is understanding, using the extensive toolkit that's been emerging and that continues to emerge, the function of the non-coding variation in the genome. Without question, we are still a long way from having this understanding, but we build it up piece by piece, technology by technology. I think, we're, many of us, convinced that sort of baseline EQTLs don't really provide the answer to too many of our, profound questions. Maybe once we see serum proteomics at scale and many other versions of these technologies that involve single cells, that involve spatial orientation of cells, we will have opportunities to get further down this. But nonetheless, there's a tremendous toolkit that's being built that can be deployed here. And whether or not we do that as a single project or many diverse projects, I think is one good topic that we're going to hear a lot about at this meeting. That said, I do have to point out that, of course, we're today not very good at deploying these techniques and databases of results from the techniques thus far in the service of this very specific question: can we take a non-coding variant implicated by GWAS and pinpoint the gene? In fact, a really nice paper very recently at the end of 2021, published by the group at Open Targets and many who are likely here in the audience, did a really nice job of dissecting this problem and came to the conclusion that, in fact, the most prominent predictor of what the responsible gene would be was still, at this point in time, despite the incorporation of large sets of molecular QTLs of various flavors, despite the incorporation of tremendous amount of chromatin interaction data, was still proximity to the gene. And so clearly there's room to grow while we are getting better at these activities. That was made even more clear to us. Wei and Kristen and the entire, practically the entire human genetics community involved with biobanking looked at a new asthma GWAS of 150,000 cases, which produced 180 hits. And this is a manuscript on bioRxiv that you can have a look at to go through the results from this study, and applied a number of distinct computational techniques, each of which had certainly good properties to recommend their use. And in the interpretation of those 180 hits, what you can see here in this upset plot, I really like upset plots as in comparison to Venn diagrams of the past. But what you can see here is that the vast majority of genes that are nominated by any one of the computational approaches that utilizes those molecular data along with genetic information are nominated only by one of those approaches, which means that for the vast majority of the 180 hits, most of these are clearly pinpointing the incorrect gene, because otherwise they would have to, by definition, agree with each other more often. And so that also, continues to give us pause. And I think basically doesn't say that we'll never be able to do this, but it seems today's underlying data doesn't permit us to do this terrifically accurately. And I think we need to, think about that in terms of prioritizing what it is that we're going to do as a community, even if we don't formulate a single monolithic project that will somehow solve this. Now, on the other hand, we have over on the left my sort of favored part, because on whatever percent you want to ascribe there, I'll just say it's 10% now, the GWAS has pinpointed post-fine mapping what is likely the causal variant and therefore the causal gene, because it's a missense or otherwise disruptive variant in a specific gene. Moreover, what we begin to see is, as sequencing has begun to become cheaper, and as the scale of sequencing in both disease consortium activities and biobanking activities has begun to increase, we see more and more examples where, while the GWAS has pinpointed a non-coding common variant, the sequencing has identified independent rare variants. And in this case, we then have a collection of variants that can be ascribed and connected to a single gene as an entity. And we can move more genes from the left to the right or the right to the left, depending on which orientation you're looking at the slide. And this is powered by a couple of things. One is that more diverse populations and founder populations, such as Finland, Iceland, French Canadians, Ashkenazi Jews, which have outsized and unique rare variant profiles, allow us to discover these additional associations, as in the example that Masa had in the earlier slide, as well as the fact that exome and genome sequencing is now reaching scale and finding rarer coding variants that escaped the detection of the early GWAS studies. The paradigm of analysis is the same. It's simply that we can apply it in diverse populations, we can apply it to rarer variation to find more clues, and those clues coalesce often at the same gene and target it in a much more precise and actionable way. So I think a lot of interest in human genetics, at least from the industry side, emerged from analyses early on, such as this one by Matt Nelson and colleagues at GSK, that I'm sure people have seen hundreds of times in slides, where the key lesson was that evidence from genetic studies, whether it came from a GWAS or whether it came from a Mendelian disease, was very influential in improving the odds of ultimately getting a successful therapy through to market. But the challenge, of course, is that this starts with the presumption of a gene being implicated. And the challenge, of course, is that the GWAS isn't always giving us clearly pointing to a gene. Wonderful. So back to where we were in IBD 5 or so years ago, the genetic studies in IBD had been successful. We had deployed a high-resolution genotyping reagent of the immunochip to perform a fine mapping, attempting to genotype just about every variant that we knew existed at the time in 100 such regions, and nailed down, the responsible variant in a few cases, or a very small credible set of variants in many other cases. We could see the strong enrichment of coding variants, also a nice but more modest enrichment of variants that disrupt transcription factor binding sites. But what we saw that was sort of most influential in a sense in the thinking going forward was that when the genetic association discovered sort of agnostically by GWAS was eventually fine mapped to a coding variant, there was a much bigger range of effects available. Again, nothing at all surprising about any of these observations, that GWAS results would be enriched for coding variants and that those coding variants might have stronger effects. Nothing surprising, but what it sort of how it influenced us was to think that maybe we need to invest a little bit more in this sequencing, which I anyway had sort of avoided in its early days as applied to chronic disease, based on a lack of evidence that rare variation was necessarily going to play a large role in common diseases. But it was clear that in the cases where we could discover those, not only were we getting clear actionable pointers that could be taken forward experimentally, but these variants were having a more substantial individual effect than those that we were discovering that were common and sitting in anonymous non-coding regions. And so there have emerged, and there continue to emerge, as I said, as we push forward more sequencing data, more GWAS studies in diverse populations, and especially as the technology for imputation from existing array data reaches to lower and lower allele frequencies because of much better and deeper sequencing references. So I give one example just from the IBD work as to how these observations can be taken forward in, I won't say real time, but in a realistic amount of time. And this is a long-standing partnership that I've had for 15 years with Ramnik Xavier that I'll draw on a few examples from through the talk. Manny Rivas, when he was working with my group back now, a decade or so ago, identified in some of our earliest sequencing data a very interesting splice variant in CARD9, which causes premature truncation of the protein and strongly protected against the development of IBD. So influenced by early examples such as that from PCSK9, we thought, hey, this is a great example. Maybe there's lots of these protective truncating variants around, and we should really dig into them. And so, of course, Ramnik took up the task, and bringing it into the context of many of the other IBD associations that we had been discussing with him over the previous 5 years, it was clear that this was another in a category of essentially microbe sensing pathways that was being implicated. Though in this case, what was unique and interesting about it was that unlike most of the others, which pertained to bacterial or viral infection, this one clearly marked a pathway that was involved specifically in fungal response. And this was well known before we, of course, found these variants responsible for Crohn's disease, both a slightly overactive common coding variant, which originally pinpointed the gene from GWAS, but this end truncation in which a functional protein is made, but it's simply missing the C-terminal domain, but it is otherwise functional, which is a good thing in the case of IBD and our research, because it was well known by that time that frank loss of function, especially in recessive form, give an extremely high penetrance and very risky susceptibility to candidiasis and really general immunodeficiency against fungal infections of all kind. And so the end C-terminal truncation doesn't seem to provide any such risk. It's not a loss of function mutation in the pure form. It's simply losing some function, which in this case ends up allowing us to observe a protective effect in IBD, but no consequent risk to any infection when we look across data sets such as the UK Biobank and FinGen. So Ramnik's group moved forward over the next couple of years to really dissect and understand the precise function, not only of CARD9, but specifically of the C-terminal domain region that was not nicked off, and identified through proteomics experiments that TRIM62 binding was key to ubiquitination of CARD9, and that the TRIM62 binding site was the key element that was missing in the C-terminal domain that was nicked off. That, in fact, ends up with a positive effect on inflammation and response that then his group set to attempting to mimic with small molecules, understanding precisely that what needed to be done was block this one protein-protein interaction, not take a blunt hammer to the whole system. And they were able to do that by cleverly setting up assays to identify where a compound might specifically impair the interaction with TRIM62, but leave the other functions and interactions of card9 definitively intact. And they moved forward, and only a few years later were able to identify lead compounds that might do that. Now, that brings us to a point where clearly at some point, the research community, the academic community, can only take this process so far. I think that's a great example when you get to the point of lead chemical matter and cellular models in which to test it. That's a great point for a transition to a more industry approach to ultimately delivering a medicine. Where that transition point and where the traditional roles of academic research and pharma take over, I think would be a great topic for discussion, given all of the parties that we have here at ICDA, not just today and tomorrow, but on an ongoing basis. And I hope that's one area where we can really build out with the Pharma Council and so many academic contributors here in the discussion, how we better sort of identify and staff the interface between these activities. What it also, pointed out to me, and we have a number of other examples like this, was that this was possible to do in only a few years because the genetic study allowed the fast track to be taken, that definitively we could identify a gene and a specific perturbation. And because that specific perturbation had a specific and easy to identify molecular action, it could be carried forward very rapidly and tested directly in a variety of cellular models. And that is, of course, in a sense, what we need to be able to do more systematically. And so the way, at least that I've formulated, and I look forward to sort of hearing how other people formulate this, is that a lot of the activities that we discuss are in terms of identifying and understanding the molecular patterns of gene regulation and how we interpret the non-coding genome is really an activity to generate a hypothesis, to ultimately generate a hypothesis of a perturbation, in this case a non-coding one, and in what context it acts and on what gene it acts in order to influence disease risk. Whereas some of genetic discoveries clearly come built in with that information and can take the fast track, but ultimately they get to the same place, and the types of cellular models and bespoke assays, such as in the card9 example, can be deployed more systematically. But there's not really likely any sort of comparable fast track to the physiology and medicine part. I think there's still always in these activities, such as I've seen anyway, the need for the careful molecular work that's done on the appropriate cellular models with the appropriate assays and readouts. So jumping back to the genetics high level, and I just go through a few points quickly. The genetics activity in IBD turned to exome sequencing primarily, now a little bit of genome sequencing, but mostly focused on the exome sequencing. We've, along with the Sanger Institute in the UK, performed a massive amount and been actively sharing with each other the data from these sequencing activities, which we think by the end of 2022 will come close to having examined close to 100,000 IBD cases in exome or genome sequencing. We have recently posted a manuscript together with the entire International IBD Genetics Consortium, which shows how, in fact, you do see both novel genes emerging from the sequencing activity, as well as genes that had been indirectly indicated by non-coding variation, as in the earlier cartoon, having independent, rare, and low frequency variant implicating risk. We also, in addition to univariate analysis, deploy standard burdens. This is all very standard, so I'm not going to bore you with any of the technicalities and the details, but you find examples both of burden tests that give positive indications for a gene, as well as individual variants. And in the case of IBD, it's much more often the individual variants that speak for themselves. In the case of other diseases, of course, it's much more often the burden test that gives you the pointers that you're looking for. So of course we run both and both. And at the end of the day, where we've gotten to now in IBD is a very substantial number of clues, many of which, those in yellow and red, pinpoint coding variants and therefore have the opportunity to take the fast track into molecular modeling. And many, many hundreds, of course, do not and still need much, much more interpretation. So not all genetic architectures are exactly the same, but they do, as far as common diseases go, share more similarities than dissimilarities. IBD is particularly productive in terms of delivering the coding variants and allowing these types of experiments to then proceed further. My good friend Romnick has, of course, taken now probably almost close to a dozen of these through a similar trajectory of cellular and mouse models to understand the role of the gene, what happens when it's knocked out, and then what happens when the alternate form that's been discovered in the genetic study is put back in as a systematic way of understanding the role of these genes in disease. Now, as I said, not all architectures look the same. And so we've been in parallel with Ben Neal and many, many others in the psychiatric community, working especially on the sequencing studies of a very, very similar scale and nature to the IBD ones in schizophrenia. And TJ Singh has led this work in our groups in Boston. And the picture here is slightly different. Actually, it's kind of definitively different in a couple of principal ways. And I put them up side by side with the x-axes now aligned to each other. And what you can see is that we have discovered a lot of things in a low frequency range, and particularly interesting and exciting coding variants that we can pursue experimentally in IBD, and have discovered almost nothing of that nature in schizophrenia. Whereas in schizophrenia, you've got extremely rare and often de novo mutations piling up in individual genes, quite similar and mirroring the initially arising copies that have been identified. And so you'd be entitled to ask two very relevant questions of this picture. One is, where is the ultra rare stuff in IBD? And I think that's the easier question to answer, which is that you only get to an IBD study if you do not have a pediatric immunodeficiency, of which there are probably 50 plus known that have IBD as a component. And so those individuals simply aren't in our sequencing study, but they surely exist. And that's a well-established part of the architecture, just not in what the common studies of adult onset or even late pediatric onset IBD look like. The missing piece in schizophrenia is a consequence of natural selection. And that is that because of the very strong force of natural selection against any variant that even confers 50% or doubles the risk of schizophrenia, we simply have not performed enough genetic studies of any kind at a sample size that would allow us to find those intermediate effect variants because they're simply kept too rare in the population. And so if we go back here, it's not that, these pictures tell us, in fact, more about what our study can detect and not exclusively about what's actually there or not there. Of course, there will always be imperceptibly, weak, rare variation, which we're never going to have a sample size to find, not so interesting. But because the power of our studies declines precipitously as we go to the lower and lower allele frequencies, the discoveries that we make right now require much stronger effect alleles to be detected. Those are kept out from this frequency range in schizophrenia. Those in IBD are predominantly protein coding variants because, as the earlier picture showed, the protein coding variants have the opportunity easily to have much stronger effects than we customarily see otherwise. So to answer the sort of my assigned task today, which was to sort of discuss a little bit about my opinions on where human geneticists should go next, I pose this as a question only because, my opinion is only one. And I think looking at the agenda for the meeting today, what's great about it is that almost everyone, to a certain extent, is addressing this question of where does human genetics go next to be maximally productive for humanity as a whole. And there's many different ways and directions and opinions on that. So I hope everyone takes up the charge of addressing this. I'll just give a few, brief ideas before closing. A few things that I think we've overlooked, and that's what I'm going to focus on just in the last, section. One is I think there's a tremendous opportunity to better understand pleiotropy. And I think, and what that allows us to do, in my mind, is potentially better triangulate and understand the shared biological processes between disease and really get at the molecular underpinnings of disease at a more fundamental level. And the reason this has been overlooked is quite naturally that most disease consortia work on one or a very close family of diseases and are not broadly sharing and intermingling with many of the other disease consortia. We can see the value as we start to look at biobanks at scale. This is an analysis from Massa and Jacob working with Hillary in the UK Biobank, and see even just by studying 100 some odd traits in the UK Biobank, you see a huge number of associations. And they start potentially to form patterns where a common set of seemingly unrelated diseases is associated to a variant on chromosome 5, a variant on chromosome 8, and so forth. And those can start to pinpoint and describe much more precisely biological processes underneath them. Favorite example of mine that I'm sure people are bored with me talking about now is a metal transporter, SLC39A8, which has one of the most pleiotropic, presentations across human genetics literature, lowering risk to Parkinson's and blood pressure, but increasing risk to a wide range of things, including my two favorite phenotypes, schizophrenia and Crohn's disease. And the transporter transports a variety of metals, but it's been clear for many years that the A391T variant that we're seeing is associated to all these phenotypes is deficient specifically in manganese transport. It's been associated with lower serum manganese levels in human. And the Mendelian deficiency is well described and severe for this gene. What Romnick's group has done recently in a PNAS paper 2 years ago was demonstrate not only the functional consequence of the mutation in the mouse gut with respect to its mechanism in Crohn's disease, but also demonstrating that in this mouse model, you can recapitulate the phenotype of the A391T variant with manganese deficient diet, and similarly go in the opposite direction, opening up what was hoped for as a possible, or giving a lot of strength to the idea that this might have provided a fundamental insight into how manganese supplementation might be utilized to counteract risk or increase risk depending on what your phenotypes of high risk are. And so we've taken not only that example, but started to look more systematically in FinGen at these collections of phenotypes to identify the idea that there might be patterns seen across phenotypes repeatedly at distinct loci, and that those might begin to group the loci. And thanks to Eric Fallman at Pfizer for sort of spurring our discussions in FinGen towards this end and proposing a volcano plot that is a nice, to me, very effective way of visualizing the diverse outcomes of a single SNP in the genome. And a number of such examples begin to elevate hypotheses of interest linking cancer and chronic diseases in opposite directions. I'm not going to spend any time on the sort of most obvious of these, discovery opportunities. I think it sort of goes without saying that the same discovery paradigm that's served GWAS, consortia so well and biobanks so well simply has not been applied deeply with respect to rare variation that's sort of below the level of what imputation can bring back. My expectation is quite strong that because we will only be discovering the strongest such variants that are possible in these lower frequency and rare range, we're going to see a much higher proportion of coding variants as we see in Mendelian disease. And much more can likely be done to synthesize, and this is probably more a computational task that we might discuss and take up as a community, to synthesize the common phenotypic insights that begin to emerge from biobanks and disease consortia with those from Mendelian disease activities that have been ongoing for many, many years now. Diversification is, again, obvious and I think critically important, both from the ethical standpoint and from the scientific opportunity standpoint. This is probably first on the list. I think we have seen many plots outlining the sort of imbalance, the sort of diversity problem with genetic studies, and I think it's not that much of a reach to say that that simply in a way is mirrored by or mirrors and is a consequence of the economic disparities in our world. And there seems, it's hard to argue, where the investment is coming from being in sync with the biases and that we see in the outcome in genetic studies. But the scientific opportunity, setting aside the obvious, ethical one, is that we are frankly missing a tremendous amount of variation. And if we simply ask the question of, gee, these coding variants are nice, can we get more of them? Well, yes, the answer is very much. If we consider that we've tested, let's say we've tested well variants above 1 in 1,000 frequency in European or East Asian populations, simply by performing similar studies in African and Amerindigenous ancestry individuals and looking at only variants not in that well-tested set, we would immediately almost double the number of coding variants examined. And this is before we even go into the opportunities provided by diverse and isolated populations around the world, populations with bottlenecks that bring forward at high frequency deleterious variants, such as in Iceland and Finland and so forth. So, and then if we go even further down the frequency spectrum, it's a no-brainer entirely that there's vastly more variation than has been examined, there's vastly more variation yet to be examined than that which has been examined. And so even if we understand, thinking about, there might be clinical uses of genetics and they'd be imbalanced. And I think we could have a great, discussion. I'm sure we'll hear later from others on sort of whether or not PRS scores are clinically useful. And that's a great debate in and of itself. I think it's rather inarguable that as currently constituted, they are so much more informative in European individuals. It's to me almost as if someone, you went into the doctor and they said, let's use the PRS score, it would be as if that depending on the color of your skin, the doctor ushered you into one room and you had the 100-year-old blood pressure machine, and in another room you would get the modern one, and somehow expect that the diagnosis of hypertension or the assessment of risk or the prescription would somehow be equal. And I think we know that that's, the latter is laughably silly. We couldn't imagine something like that. And I think we have to sort of wrestle with the fact that we need to do a lot better to make sure that this is not the case with respect to genomic tools that get deployed. And then finally, the last one of these mini vignettes, because probably I'm boring people by now, is I think disease progression genetics has been vastly underexamined. When we think about the fact that most therapeutic development is targeted at building a medicine to slow or arrest disease in individuals who are already sick with that disease, it seems that we're, in a sense, really missing, or misdirecting our efforts in some cases to not sort of more earnestly take on the challenge of looking at disease progression genetics, because it might tell us some new and different things. Now, it's hard. Technologically, there's been a lot of effort put in by many. It's a paper I cite just by Jihong Lin and Wei Zhu and Runak Day, working with her, developing an efficient and powerful genome-wide approach that's to survival analysis that can be deployed effectively in progression studies. We've started to do this in FinGen, but the scale is a real challenge. And I think we need to wrestle with the fact that the technology for doing this, we know how to do it, but in few places, I mean, one of the many reasons I've been spending the last 4 years in Helsinki is that the availability of longitudinal and lifelong data in Nordic registries is a powerful tool in this dimension. But the scale of such studies, requiring that you start with disease A and then move on to disease B or complication C or what have you. And then the lack of collaborative efforts in this area, I think, are really holding us back potentially. So to finish, I guess the question we posed was, is there a foundational activity such as the Human Genome Project that we could work on together as a community? And I think if you read that in a simplistic way, the answer is clearly no. There's not a foundational project that's going to unlock everything, because it's extremely complex. And I think there's diverse pathways. We have to be prepared to invest fully in the long term in understanding the biology of the genome, and also invest fully in the immediate term in harvesting as much as possible for the fast track of coding variants that can go immediately into effective therapeutic hypothesis development. So there are many things that need to be done in parallel, and I think those need to be done collaboratively. And that's, what I think we've been able to accomplish in terms of coming to a conclusion to that first original posed question over the last 4 years. So many thanks to many, many people who it's not really even conceivably possible to list here, but especially my long-term partners in the International IBD Genetics Consortium, in the FinGen project, throughout the ICDA, in the PGC and Schema consortia that have made such enormous progress over the last 15 years in psychiatric genetics, and the NOMAD project, which is near and dear to my heart always. And especially Ramnik, Ben, and Arno, with whom I sort of have worked most closely with over the recent years. So happy to take any questions, and welcome to the meeting.
Amazing. Thank you, Mark. Questions are coming in on the Q&A, and I'm encouraging people not to be shy. Masoud, I hope you agree that we have already addressed your question, or Mark did, rather, in his talk, where he asked, are we identifying too many susceptibility loci in GWAS and not so much associated with disease progression, the latter being more relevant for drug target identification to treat patients? What can we do more in this regard with current biobanks, including FinGen? So I think you already answered that, Mark. So we're going to hop on to the next one, which is coming from Nabila Boyata Naji. And if I butcher somebody's name, please bear in mind that I'm speaking Swinglish and it's not intentional. Thank you, Mark, for this great talk. Any thoughts on how to deal with potential therapeutic leads from pleiotropic loci with risk to disturb key processes involved in too many important biological functions and maybe compromise targeting them to treat disease?
Yeah, I mean, I think it's actually, I didn't state it, but I think it's actually one of the most important reasons that we have to think along these lines and really squarely address pleiotropy. And that's one of the, I think, perhaps the most valuable contribution that biobanks are able to make is to look across diseases. And so I think it matters tremendously whether you're seeing, as in some of my graphics, both strong risk and strong protection to different diseases coming from the same variant, or whether there's a uniform directionality of those associations. If we know what they are, it's possible to mitigate them. And if we know what they are, we can, consider what we need to look for, clinically, consider whether the timing and severity of the diseases is consistent with pushing forward with a therapeutic hypothesis or with it being a non-starter.
Great, thank you. And then we have Anonymous, who has asked, how do you think ICDA can help diversify genomic studies and data to study underrepresented population with all outfalling into scientific colonialism and support underserved populations?
So it's a great question. I think we need to, in my mind, I think we need to address this in formulating not only study designs, but also funding models. And so one thing that we think about is, how do we balance the desire to move quickly with available data, which is biased, and at the same time make the appropriate investment in diversifying and supporting biobanks. And I think we need to commit to, when we seek funding for all of these activities, to ensure that all of those components are funded. And so if someone comes, let's say, industry decides they'd like to fund, a big genetic study of phenotype X, and we have lots of phenotype X in, well-developed biobanks, then we have to be of a mindset that the funding for such an activity has to explicitly have, as a component of it, funding that goes to the development of biobanks in parts of the world where there has not been that investment, in order that any activities that we undertake as a community are, whatever their activity is, supporting that mission to build and diversify the genetic landscape.
I agree. And also to comment on that, this is a topic that we'll come back to later in the scientific program. So I'm encouraging Anonymous to stay on his or her toes and follow the discussion, because we do have a commitment and representation from all continents, not all countries, but all continents on ICDA organizing committee. And we're thinking a lot about diversity and doing that in an inclusive and sort of equal way as much as possible, both on the science level, at research participant level, and also scientific output level, because of course we want to find solutions that benefits everyone, and that has to bleed through the entire scientific process, if that makes sense. So I'm encouraging Anonymous to stay on his or her toes and keep on asking the great question. So we have time for, I think, maybe one, maybe two questions more. So Cindy Lawley has asked, cohorts often struggle in a trade-off between putting resources into higher enrollment versus longitudinal samples. I think of this as a breadth versus depth trade-off. Is there anything you can share about how you think about this, Mark?
Yeah, that's I mean, it's a constant struggle, and I don't think it's not even restricted to biobanks. I think all of our studies, disease studies alike, have the same tension in that we've recognized that there's a huge scale that's required for the genetic discovery. However, once the genetic discovery is made, you find yourself greatly wishing you had a much deeper clinical phenotypic and longitudinal development of data around those discoveries so that you can interpret them. For me, that's, there's a few primary reasons why I, relocated 4 years ago to the Nordics, and that's one of them, the opportunity to have lifelong registry data available for research. And so I think there's certainly that opportunity existing in some places. I don't think there's an easy answer to the question, because it really depends on what your priority is in terms of, if you're building a resource to discover genetic variants in a disease that has not had much discovery, then scale, I think, has to be of prime importance. If you're interested in a phenotype, for example, something like IBD, where there's been an inordinate amount of discovery, the premium is perhaps much more in the other direction of, do you have a sufficient depth and length of follow-up on individuals to really understand the role of these individual variants as they're being discovered? So I don't think there's a sort of a one-size-fits-all answer to this, unfortunately.
So great. So with that, we don't have time for the final questions, but I'm encouraging, as I did in the beginning, everyone to lift over their questions into Slack and continue to have an active discussion there. I am confident that everyone in the ICDA community want to thank you, Mark, for an excellent talk and for a really sort of thorough thought through on the journey we have both behind us and ahead of us. So with that, thank you. And we're going to have a 15-minute break now. And I hope to see you all back for the next session in the Maps to Mechanisms to Medicines program we have ahead of us today in 15 minutes. So thank you so much.